Okay, I switch to English, I think, a little bit because my friend Stefan do not understand a lot Spanish, okay? Hello to everybody. In a few words to introduce now, Stefan is a great pleasure, a friend for a long time. After university studies in Nice, Stefan moved to the north of Europe in Sweden at the Chalmers University to do 3 years of PhD in Chalmers, and he still maintains a strong collaboration with Swedish research ever since. And he obtained a position in Lyon, I think in 1997, assistant professor, and he's promoted now full professor. He worked in ENS École Normale Supérieure de Lyon as a professor in this lab. Stefan is originally a chemist in organic molecular chemistry, and he evolved during his career to the chemistry of hybrid materials by choice and pleasure, he said to me. Good evolution. He currently works now in the synthesis of hybrid material, and also inorganic nanomaterials by soft chemistry, chimie douce in French, and colloidal chemistry, with also a problem of interface, if low interface problem, and the targeted studies of this material are optics, health, and environment. Stefan is a deputy director of the Lab of Chemistry at the École Normale Supérieure de Lyon, and in a few weeks now, he will be the head of this laboratory. In a few weeks also, he will be introduced in the Nanoandes network. Welcome to Stefan, to the Nanoandes network, and also with all the colleagues of the Lab of Chemistry of Lyon. Welcome to us. Now, okay, please, Stefan, you can present to introduce all the activity of your lab, and I think we are in perfect coherence with target and the domain of Nanoandes network. Okay, I will give an inform 3 minutes before the end of your presentation. Okay, 25 minutes, please.
Thank you, Alain. Very nice introduction. First, I understand Spanish, so yeah, I cannot speak, but at least I understand, so I can follow the talks and the discussions, even if sometimes it's going a little bit fast for me. But first, I would like to say how happy I am to be here and to have been invited to this wonderful network and collaborative network. It's a great pleasure, it's an honor, and for the whole group and the whole lab, I hope we will have very nice interaction, and I'm sure about that. So it's my first participation, and again, thank Alain, all the people from Grenoble and from South America that allowed that for us. So the idea here is just to introduce our research areas and to give you an overview of what we are doing in Lyon and what we can do, or where you can interact with us if you are interested. And you will have a more focused talk tomorrow by my colleague Frédéric Lerouge on the health application. So I have to share the screen. Yes. Yeah. So do you see my presentation?
It's okay, it's okay.
Yeah, I put it in a full screen like that. It's clear?
Yeah, perfect.
Okay. So we are at the chemistry laboratory in Lyon. So it's a lab which is common between ENS, École Normale Supérieure, CNRS, and the University of Lyon. You have an overview of the building here and the main hall entrance of the building. So where is Lyon if you are not familiar with France and our geography? So Lyon is here, not very far from Alain, which is in Grenoble, maybe not today, a little bit more in Chambéry, but so Grenoble is not very far from Lyon. It's less than 1 hour by car. And we are very close to the Alps. I know you have the Alps, and it's certainly a wonderful place, but the Alps are not so bad also. So you are all welcome to visit us and have a look at that. So we are not very far from Switzerland and about 2 hours by train to Paris. So Lyon is very famous for history, and it's a 2,000-year-old city, which is classified by UNESCO as a World Heritage. It's a very beautiful place. It's where the cinema was created for the first time by the brothers Lumière. It's the city of Ampère and other important people. And most of all, it's the French capital for gastronomy. So if you come to Lyon, you will enjoy very nice food. Not now because restaurants are closed, unfortunately, but hopefully in a few months it will be open again. So if I come to the science and chemistry, it's also a very important place in France as a chemical area. It's the first leading chemical producer, in France. So we have all the, I'm sorry about that, I have some instability. So it's the first industrial research center in France, in particular in chemistry, with all big groups having the research and development in Lyon. It's called Vallée de la Chimie, so it's a chemistry valley in France. You have more than 6,000 chemists working in Lyon. 1/3 is dedicated to research, with important applications and development in catalysis, material science, processes, and environment science. And what important thing also is the, in particular in this period, it's the #1 world producer of vaccines, with all big vaccine companies producing here, actually very close to École Normale, because we are located on the biotech campus here. And you see that we have about 500 research laboratories and 30,000 researchers in this area. So very important in France, very important place. If we look at the lab, it's not a very big lab. It's about 120 researchers, all included with students. So we have about 50 permanent staff in the lab in research. And it's divided in 3 research departments. The first one is dealing with modeling approaches. It's a theoretical chemistry department. The second one is dealing with more molecular, supramolecular engineering and chemical biology, interfaced with biology. And the last one, which is actually, is functional materials and photonics department, and we are dealing with materials and functional materials, hybrid materials, and so on. I will develop a few aspects of our research. The lab is funded by both public and industry and defense agencies and partners. So you have a list. I'm not going to go through that. We have an important international network of co-workers and partnership, as you can see, even in South America, but not enough, probably. So I hope to develop more within this network. We have some interaction with Brazil. So this is one of the objectives for us, is to interact more with you, and hopefully we will do it. Regarding the research expertise, so as you can see, it's quite broad. Even if the lab is small, you have seen the departments, it's broad. We go from modeling molecules to materials, and with strong expertise in the interfaces of the system. So if you look at molecules, they are engineering for optical application, for biological application, for self-assembly and supramolecular engineering, chirality. In material science, we are dealing with inorganics, with different type of inorganic materials, metallic materials, and hybrid system. We have activities in ionic liquids, and also in shaping and microfabrication, 3D printing. Of course, we are very much interested in the interfaces of systems and dealing with colloids approach, nanoparticle surface chemistry, and a different type of composites and material shaping. And regarding the modeling aspect, again, and you will see it interacts with all other fields. We have people dealing with modeling of molecule structures and the structural properties, interaction, and the relationships with biological system, DNA and DNA damage, and so on. And also, and very important, is all type of interfaces for catalytic or optical application, which means that people are very interested in molecule surface interaction, for example, how it affects the property of the molecule, how it can affect reactions that are nearby the surface. And we have also developments in ionic liquids and confined fluids in the theoretical approach. Regarding the experimental platforms and equipments we are quite well equipped for both molecular synthesis and material science with synthesis laboratories for any type of synthetic approach. We have a clean room, which is standardized for material preparation in particular. And we have different equipment for material processing, from molding to 3D printing, thin films, and so on. And you have a list of equipment. I'm not going to go through all that. You can have the slides. I don't know if you give the slides, but you can have it if you are interested to interact on one of these type of equipment. It's totally open, of course. And what is quite unique, we have two big supercalculators. You have seen that we have a department on modeling that allow multi-scale approaches in modeling with very, very strong potentialities in these different fields of from molecules to materials and interface. We have also developed a platform for 3D printing together with a university and ENS and two other labs, ICBMS and Matéis in Lyon. We have this platform, which is very interesting because we have all type of 3D printers located on this platform. And we can do any type of technology of printing, from additive printing, stereolithography, and two-photon printing. And with different techniques, we also develop the chemistry, which is quite unique. So we play both with the technological development of the systems and the chemistry. And due to that, we are able to build any type of materials by these different technologies, from polymers, ceramics, hybrids, and biomaterials. So it's quite versatile and quite broad. And you have a few examples of ceramics that have been prepared here. And just to illustrate what can be done, we are able, by these technologies, in particular two-photon microprinting, we can go to resolution that are up to a few tenths of nanometers. For example, you have zirconia structures here, that have been printed, and you see the size of the structure is about 20 micron per 20 micron, and the resolution of the material that is printed here is about 50 to 60 nanometer. So we have a nanometric resolution in printing, both of polymers and ceramics. Here it's pure zirconia in this case. And you can make surface patterning with photonic crystals or any type of network. And you have, for example, here optical response of of lenses that have been microstructured at the surface using zirconia patterning with this technology. So if I come to the different type of research project and connected application that we are doing, the first one is investigating light-matter interaction. This is a very strong research area in the lab, both on the molecular part and on the material part, but also on the characterization. We have physicists in the lab that are developing optical setups for characterization and development of these problematics. So the applications are in sensors and filters, in laser, lighting, and also development in different type of photochemistry and photocatalytic problems. And you have a few setups that have been investigated here. I'll give you just a few examples of type of materials that we can design and study in this light-matter interaction. The first one is the synthesis of luminescent particles. Here, these are YAG nanoparticles. I don't have time to develop the whole synthetic strategy, but you have here a dispersion of YAG that are doped here with cerium. The dispersion here is about 50% in weight in water. These are very small particles. They are prepared using solvothermal technologies, reactions. It's a combination of soldier process in high pressure and temperature. And we can prepare highly homogeneous nanoparticles of small size, between 5 and 10 nanometers, for example. And with this particle, we can play and we can make them react together, make gel, for example. And from the gel, we can make xerogel or aerogel by supercritical drying. So aerogels are porous, xerogels are dense. This depends on the type of drying you use. You can make films also. And what is interesting here is just the nanoparticle, which is here doped with cerium, so it's an emitting nanoparticle. You can mix different type of particle together. And so you can make a pure gel of YAG, or you can make also a mixture. You have examples here where we have mixed fluoride particles that are doped with terbium, with cerium, and we have here indeed 3 types of particles doped with terbium, cerium, and europium. You can mix together and make a homogeneous material, 3D material, which preserve the optical emission of the system. So you can then excite either one or all of the particle together and get, for example, here white light emission by combining the different emission. And it's a very homogeneous, since we have a reaction in solution. It's like a reaction between molecules, but between particles. And you can build all type of system by this approach. We have a lot of big activities also in metallic nanoparticles in design and just controlling the growth. These are gold nanoparticles, so we have developed strong activity in the process, trying to control perfectly the growth of particles and understanding the growth by combining theoretical approaches and the chemistry. And we have been able to develop new processes that allows to make highly concentrated solution, which is quite unique, of particles that are totally monodispersed, highly pure, and in a quantitative approach. So these are very efficient routes. It's a growth-mediated approach where we can build all type of particles just by changing a little bit the condition, the reducing agent or the stabilizing agent. We can make all type of particle. Here is our pentatwin-based structure. All are gold, and you can see you can tune, and all are very homogeneous in morphology and in size. And this is important because then we are interested in their optical properties. I mean plasmonic properties. Sorry, and you can see we can tune the plasmon. So this is the absorption of a particle depending on the morphology and the size. It's very sensitive. So we can tune very precisely the optical response and the optical absorption of the particle just by changing the morphology or the size of a particle. And that, just changing some aspect of the process, you can easily control the shape and size of particles, and thus the optical response. And then we use them in different projects for Earth, for optics, or for photochemistry. So this is a strong part of our activity. And you have an example where we use this particle in sol-gel material to produce highly efficient optical filters that are used to protect sensors against intense radiation, lasers, for example, just by combining a molecule and one of these particles, selecting the appropriate shape and size to correspond to the wavelengths we want to address. And you can make a material. Using sol-gel process, you make silicate-based materials with a very high performances in terms of optical protection. And we can also play with this system from the visible or the visible area to the near infrared up to 2 microns. And this is extremely interesting in terms of optics, because it's not only the visible, but also the telecommunication wavelengths that we can address with this approach. Second type of activities I'm going to show you is our developments in the field of environment and energy. So we have a lot of activity, in particular in modeling on catalysis and catalytic approach, development of mesoporous material for catalysis, bio-inspired catalyst, but very important in silico design of catalysts. So we have a lot of people involved in the design of catalysts by theoretical approach and developing methodologies in this. We have activities in energy also, mostly in batteries, by designing either electrolytes, polyelectrolytes, or electrodes, which are inorganic, lithium-ion, for example, based electrodes, and modelization and modeling of all interfaces in batteries. And the last one is related to environment. We have activities in water and air treatment by using, in particular, photocatalytic system that we developed, hybrid coatings that we developed and can be applied on different type of substrates. Just to illustrate that, you have a work here that was done on lithium titanate that is used as electrode in batteries. And it's designed by the same approach as the YAG that I've shown previously for optical application. So the synthesis of a particle is exactly the same route. It's a solvothermal route that allows the preparation of very small particles that can be reacted together to build hierarchical structures. And here you have a hierarchical porosity building of a film. So we make a film that will be used as an electrode. And why it's interesting to have a hierarchical porosity is to improve the lithium diffusion in the electrode, to try to improve strongly the capacity and the efficiency of the battery. And it worked. And we have an extremely efficient system here, where we can rapidly attain the maximum capacity of the battery by using this kind of structured electrode, and with no damaging. Even after 1,000 cycles, we keep the efficiency of the battery. So it's a very stable system, very efficient, and you can load it extremely rapidly. And this is mainly due to the hierarchical structuration of the system. The last part is devoted to the biology applications and healthcare development that we are interested in. So we have activities in imaging. This is a very important part of activity, and Frédéric will talk about part of it tomorrow, in particular a combination of MRI and two-photon imaging technologies. We have development in biophotonics and scanner technologies also. Therapy, with modeling of DNA lesions, drug delivery strategies, and photodynamic therapy, always connected to light-matter interaction, and more recently, development in theranostics by, for example, probing enzymatic activities or developing multifunctional nanoplatforms that are of interest, I guess, in this consortium. For example, we develop systems where the heart, the core of a particle are built of fluoride, raw earth fluoride. Here it's a gadolinium fluoride system. Gadolinium fluoride because it's a very interesting element if you want to make MRI, this type of imaging technology. But we can functionalize it. We have designed a platform where we can functionalize with a dye that can be used for in vivo biphoton imaging. And also we can have some targeting entities. What is also interesting is by using the gadolinium, it's an element that can be used for a new generation of scanner. So Frédéric tomorrow will talk about MRI and two-photon in vivo microscopy. What I show you on this particle is what we have done in partnership with Philips. Philips have developed a new generation of scanner, which is a KH-based technology. It's a spectral photon counting CT scan, which is completely innovative because you excite only on the KH of the element, which means that we can be specific of the element in this new technology of scanner. So we are developing all the probes that can be used with that. And in particular, the gadolinium fluoride particles has been demonstrated to be extremely efficient. You have here the example of KH imaging of a rat. So this is a normal scanner that you can make in a normal scan. And this is the new technology of scanner, where you can see the difference here is that here you map everything, but here you map only the gadolinium. And what is very interesting is that you can also determine the concentration in each part of the body, the concentration of gadolinium by this technology. It's a high resolution, very efficient and quantitative technology, which is extremely promising for the next future. It's going to be in the market quite soon, I guess in 2 years from now.
2 minutes, eh?
Yeah, I'm almost finished. This is another project that is developed by Chantal Andro and Yann Bretonnière in the same area, is using this time pure organic system to aggregate them, and they change their optical properties when they get aggregates. They start to get a high fluorescence, and if you do that in the presence of a surfactant, for example, you can stabilize small nanoparticles. Here it's about 80 nanometer in diameter, so you can visualize them and use them in bioimaging and two-photon imaging. And you have example here of two-photon imaging, two-photon microscopy of these pure aggregates, organic aggregates, nanoparticle that has been injected and imaged in a tumor. You can see the vascular circulation and accumulation. You can see the tumor. There's a tumor here in the middle, and the particles are not getting inside the tumor here. Yeah. So just to conclude, in the lab, we are going from molecules to material and shaping, from the design, and we can go to the far down to the setup and even transfer technology. You have a few examples of transfer that have been done in the lab to the market in different applications. And finally, to end, I just show you that we are organizing next summer. Some of you know already, because Alain, Géraldine, Xavier are very much involved in the organization. We will have in Lyon and together with Grenoble the Soldier 2021 conference next August. So of course you are all welcome if you are interested. Just get in contact with us, and we'll be pleased. Thank you very much, both for the invitation and for your attention during this presentation. I can answer if you have any question.
Okay, thank you, Stéphane, for this nice presentation of your lab. I have a question of Sarah about
Yes, of course.
fluor.
Hello, Sarah.
Sarah, you can speak, or I can. Okay.
Okay, I can speak.
Yeah.
I thank you very much for the nice talk. I wanted to know how is removed gadolinium fluoride from the organism, because it's a nice image, but perhaps it's not very healthy.
It's today the one that are used in human are gadolinium complexes mostly. It's cage complexes, and you are right. There are problems of gadolinium release with this system that are already used. So it's a big concern. What I can say is that gadolinium fluoride are very stable particles. They are highly crystalline, so they don't release. The elimination, you will see tomorrow. You will see Frédéric tomorrow will present much deeper insight in this technology. They can, of course, you have normal elimination by the body. You can have some retention depending on where the particles are going. We have tomorrow. You will have examples of brain imaging. So we have a targeting approach for Alzheimer's disease to target amyloids, for example. So in this case, of course, the particle is staying in the place, so that we can image if you target. What is the behavior afterwards? It's too early to say. Is it toxic? We don't know at this stage. We have no insight into that. We have run a lot of toxicity tests in Grenoble, actually. There is a special lab with experts in toxicity measurement, very highly developed test. We have never seen any toxicity of a particle in any type of cells or organism. So they are quite safe on the cellular point of view, even on, as I said, highly advanced toxicologic tests. They have been shown to be safe on different type of animals until now. They are used on small and medium animals at the moment. But of course, for the moment, there is no idea to inject it in human. No? It's not the point here. We are developing together with Philips the scanner technology, and we have also technological approach on the imaging systems. But there is a strong need of this kind of particle for these new imaging technologies. So we'll see what the future will be in this sense. But it's of course a big concern. But it's a concern with all type of particles, even in other applications today. Here, of course, it's intravenous, but is it more toxic than if it's by air in a pulmonary, or I don't know. It's a very difficult question.
Okay.